Beakers are incredibly versatile pieces of lab glassware. They're cylindrical in shape and come in various sizes. While they can be used to hold liquids, they're not the best choice for precise measurements. Instead, beakers are your go-to for mixing solutions, heating liquids, and general storage in the lab. Burettes are specialized pieces of glassware designed to deliver very precise volumes of liquid. This makes them essential for titrations, where you need to add a reagent drop by drop to a solution until a reaction is complete. A burette is a long, narrow tube with a stopcock at the bottom for controlling the flow of the liquid. Markings along the tube indicate the volume dispensed. Test tubes are cylindrical vessels used in the laboratory to hold and mix small amounts of substances. They're a very common and basic piece of lab equipment. Desiccators are sealed containers used in a laboratory setting. They contain a desiccant, a substance that absorbs moisture, and are used to remove moisture from samples and protect them from humidity. Petri dishes are shallow, cylindrical vessels used in the laboratory to culture cells. These dishes provide a sterile environment for cell growth, allowing scientists to study microorganisms and other biological samples. Condensers are used in the laboratory to cool hot vapors and turn them back into a liquid. This process, called condensation, is essential in distillation, where substances are separated based on their boiling points. Balances are essential tools in any laboratory, used to accurately determine the mass of substances, ensuring the correct proportions for experiments and formulations. Centrifuges are essential lab equipment that use centrifugal force to separate liquids and solids based on their density. This process is crucial in various pharmaceutical applications, from separating cells to purifying drug compounds. Microscopes are indispensable tools in pharmaceutical research and quality control. They allow scientists to visualize minute structures, from cells to drug particles, ensuring product purity and identifying potential contaminants. pH meters are essential tools in the pharmaceutical lab. They measure the acidity or alkalinity of a solution, which is a critical factor in drug development, formulation, and quality control. Spectrophotometers are essential tools in the pharmaceutical lab. They measure how much light a substance absorbs or transmits, allowing us to determine its concentration. This is critical for quality control and drug analysis. Stirrers and hot plate stirrers are common lab tools. Stirrers are used to mix solutions, while hot plate stirrers can simultaneously mix and heat them. These are frequently used in pharmaceutical research and development to ensure consistent and controlled conditions. Gas chromatography and HPLC are crucial techniques in pharmaceutical analysis. Gas chromatography separates compounds in a gaseous state, while HPLC separates them in a liquid state. Both methods allow us to identify and quantify the different components within a mixture, ensuring product quality and purity. Titrators are specialized lab equipment used to dispense precise amounts of liquid, known as the titrant, into a solution. This process, called titration, is critical in pharmaceutical analysis to determine the concentration of a specific substance. Condensers are used in the laboratory to cool hot vapors and turn them back into a liquid. This process, called condensation, is essential in distillation, where substances are separated based on their boiling points. Bunsen burners are a common heat source in laboratories, used for sterilization, heating samples, and combustion. Enjoyed the video? Want to stay up to date with the latest GMP insights and guidance? Then make sure to subscribe to our channel. We've got tons more helpful content coming your way. Hit that subscribe button and join the GMP Guide community.